Thanks for having me, and thank you for the kind introduction. So this is like the first of all, no disclosures, and this is kind of the agenda that I have planned for today. So I discuss a little bit data analysis ecosystem, how I see the world of omics data, and then I look a little bit into variance. Searching for variants, for causative variants, and then I have like two more topics, which is really about national research data infrastructure for these topics, and a nice tool that we recently developed. So the omics data revolution is really, I mean, many times it's discussed in data amounts, but I think that is too short. I mean, if you have 1,000 genomes, that doesn't scare me, and if you have 10,000 exomes, doesn't neither. But if you have like single cell attack-seq and some chip-seq and some long-read sequencing, and you want me all to integrate that, that scares me. And this is like, I mean, a nice cartoon from Leo Pachter's website who kind of visualizes that because for all these different things, every biomedical researcher tries to transfer his question into something that he can then turn into sequencing data. And there's now this many protocols for doing that. And each needs a different kind of analysis, different kind of workflow, depends on different metadata and interpretation, especially if you move forward to genomes and single cell, gets very interdisciplinary because you really need knowledge from different domains. So that is what I think. So my slice of this hell is like the core unit, and this is like a nice figure that came out a few years, how the things, the works distribute really. So there's a few things that you do many times, and data management is one of them, but analysis wise, there's a very long tail of individual things for translational research. And so we do like 200 projects a year in very different fields: cancer genomics, rare disease, single cell, very popular nowadays, mass spec also, and of course integration. So, and then my research is really to come up with ideas and tools and methods and processes to handle this kind of efforts. So I mean, there's like tools for user empowerment, there is like automation for processing, there's software for organizing data, and on the long end, of course, there is simple, normal collaborative data analysis. So my approach to that is really, I mean, we embrace all solutions that we find, and for example, we have an in-house bioportal instance for the cancer genomics people, and we have built up an ecosystem which is really loosely coupled. So whenever every time a new method evolves, a new thing comes out, we can integrate that, and we try to fill the holes there with own solutions. The core is our data management system, which is for raw data processing, results, log files, has audit trails, rights management. So because this is sensitive data, and then around that there is solutions for the different use cases: single cell, rare diseases, cancer, mass spec. So important, I think, if you design something like that, is really, it's flexible, it's scalable, it's open, that the reproducibility can be realized, so you can track what happened to your data, how they were processed, how they were used, and of course, in the end, it has to be secure and fair. So with that more general introduction, I look a little bit into variants, specifically rare disease exome analysis use case. So I assume you have some idea of doing that, but there's many steps really here. Like, first of all, lots of consistency and quality checks with pedigree and coverages, and if everything went wrong, then the second major step is really searching and refining and looking for causative variants with lots of different filters, impact, inheritance, prioritize them, maybe phenotype type based with using that kind of information, and then eventually documenting them, interpreting them, looking out in different databases and things. So that kind of work concerned us a lot, and we really try to empower like our collaboration partners here. And this went through many iterations and finally became a software that we published in Nucleic Acid Research last year. So this is for users, that's a web application. I won't walk you through the details now, but just to get an idea what you can do here. Maybe you can look what link outs. There's link outs. You can link out to IGV, so raw data. You can link out to mutation data. You can export the stuff. You can annotate the stuff with ACMG. You can select the transcripts. You can filter for inheritance patterns and all these kind of things. So this really emerged in a very interactive way in working with different people, with different labs. And the bigger picture is that we really spent some effort to make this very interactive. So there's a star database design in the back end, and the annotation is done mostly on import, and that makes it very fast and interactive, and so people like that very much. And the collaborative features are also very popular with people. So there is a project-based and PI-based structure there, and you can share data with the people you want. You don't need us for that. You can do that in self-service. You can leave notes there. You can reanalyze the things when a new ClinVar version comes out or when a new genome version comes out, and these kind of things. So you can try. We have 2 demo versions out there in the internet. One is with the classic mode, which has everything, which is filled with some public data. And there is the kiosk version with slightly reduced functionality. So the collaborative features are missing. And basically there you can upload the VCF file of your own and can try it out. Yeah. So software is open source and free. We by now have several instances in university clinics in Germany. And there's one in Bonn, there's one in Aachen, there's one in Göttingen. And more people are looking into it. In Berlin, we have like 30 users and a lot of data in there. And so it initially started in research, but it really became a nice translational success story, because by now, everything that is done in vitro diagnostics in terms of rare disease exomes, the Labor Berlin automatically puts all the data that they produce. I mean, they have, of course, the in vitro diagnostics processing in-house, but you all know that this won't solve all the cases, and everything where there's consent, and nowadays there is for most, gets into our realm, and then is reprocessed with the latest and greatest methods, and is made available to the clinicians and the researchers again. So I mean, this is probably the important error here, that the data analysis to first entered the HPO terms and gave this sample to Labor Berlin, eventually can interact with that and try solve the difficult cases. Okay, outlook, what we are doing. So easy deployment, that was something that came with the other institutions now using it. That's mostly solved. Beyond small variants, we have a really nice solution now for structure variants genome-wide with nice annotations also. That's working nicely, and we will publish that soon. So then also for structural and copy number changes in the non-coding areas. We have established beacon-based, so genome alliance protocol-based for interacting with other sites. So of course you can look up in the big databases, but you can also now we can start to like exchange data in a secure way within this network of people that are using it, and we're looking into options how to possibly certify this also. That was like the things that are already software, but of course there are then many more things in this realm that need to be looked into. And I take a small example here from a project we did with Sabine Klaassen and Jirko Kühnisch. So looking for causative variants in certain subtypes. I don't dare to explain the phenotypes here in front of this audience. But then, yeah, burden testing really didn't work, even if you used like the newer and nicer methods with calibration of the variants and with conventional things. Then you run into the small n statistics. And we did a little bit of thinking, what could bioinformatics improve at this point here beyond the statistical approaches? So, and what we came up with is the idea really to make an algorithm that kind of mimics what biomedical experts would do. But because it's an algorithm, make it reproducible, scalable, and mostly bias-free, I would say. So, and this is based on the CUT scores that Martin Kircher, junior group leader at the BIH, developed. It's a really, really nice machine learning way to evaluate variance. So the basic idea, the 3-sentence summary would be, he looks at variance. He has a model of how mutations would occur, very elaborate, and he compares that with the differences between chimpanzees and men. And basically then the functional ones will be depleted in the second data set. And that is learned by a machine learning model, and that then produces a score for the variance. And here on the right-hand side, you can see that with that, even in this small cohorts, we can see a really clear enrichment of the high impact variance. And it shouldn't say variables, I should say variance, sorry. And I think that's very convincing solution, and it's very clear. So then a little bit of what is happening in the future in the genome field. So I'm not discussing like genome projects that we are having. We have one with radar soldiers. We do a lot with Mund loss on limb malformation, but rather on the National Research Data Infrastructure. I'm a member of this German Human Genome Phenome Archive project. So the core mission there is really to establish an infrastructure for human omics data for the German research. So it should be fair. It will be fair. It involves all the major centers. It is community centered. So that's why I'm presenting it here. So to also motivate you to get involved, it solves the legal and data use framework issues. And really long-term, it should democratize like the use of this kind of data. And of course, be integrated in the European infrastructures. So it's quite a big endeavor. 46 participants, 21 institutions. There's the directors, and it's a clear understanding within the network that the different users group, the patients, the biomedical people, the researchers, and the computer nerds have very different needs in how they want to work and interact with this data. And we try to address all of them in a reasonable manner. So this is the community. Is really community driven, and that is there's a rare disease community, there's a cancer community. Next will probably be a single cell community, but there's a clear plan how to continue and add new communities here and work with them. And here on the left-hand side, I show a little bit the Beule activities within the structure. So the communities are really guiding the process. Then I'm, for example, involved in the data processing and in the back ends, and other people here, like in Thorsten Schlomm is in the cancer field, and the biobanks are in there with Michael Hummel. And this is the timeline. What functionality will be available? So first it's archive, then it's really Atlas, and eventually interacting and in a data processing service for the whole community. So I can say, try get involved. It's now a year old. Functionality starts to grow, and I think it's good if more medical people also get engaged in steering this. Okay, looking backward, now I talked about genomes and expensive and big data. This is a small project that maybe is interesting and looks back at like data that you might have. So the motivation that we had here was there's lots of panel sequencing data that's accumulated over the years, and we are again and again have project where these one wants to make more use of this, and the real diagnostic gap is that the CNV calling is better. There's for individual panels there is one, but usually people have this huge collection from different kits, from different years, from different batches, and but they are really big. So usually there's many patients in there, so samples in there. So we kind of came up with an idea to properly scale and score this, and then call copy number variance from this. And here in the lower picture, you see a little bit the visualization of the thing. So of course, then there's bad targets like here B. There is noisy data that are then ignored like D. There is like a bad sample like C. But then there's also something here. The targets are on the y-axis. Here, then there's something in the area A, for example, that looks really interesting, and it could be like a copy number change that is systematic. Yeah? So this is the results that we are getting. So it really has two modules. First of all, we cluster by kits and batches, and then we call variance by scoring and looking at the distributions and at the outliers of the distributions to identify the copy number changes. It is sensitive and also pretty specific. So we had a test with 88 samples that we called. So we had actually more, because we also tried to validate qPCR with others from other tools called, and we could confirm more than 30 of those things. So I think that is pretty good result, and it's now under revision, and we are proud of it. So this is the team. Who should I mention? Manuel, who is the brain behind Warfish, Miko, who is, and Oliver, who do a lot of the programming, and Vincent, whose student actually is no longer on the list because he left already. And with that, I end my presentation, and thank you for your attention.
Thank you very much for those insights. So again, I would like, if you don't mind, to ask you one question. Since there's a large proportion today of our audience practicing physicians, so what is, when on your daily basis, when you need to explain and discuss your results and findings with the expert domains, not from biology, but from medicine, where do you stop having good agreements? So where does it become difficult?
No, I think it's very important that people collaborate interdisciplinarily. I mean, like in a lot of the features that are in Warfish, for example, are really ideas of people like Nadia Imke and Ute Scholl, who are working in the clinic to solve cases. And so they came up with ideas, and we made them into software. But of course, there's also ideas from us in there. But this is really the way it should be. And I think with the practical, that is the core of translation, that you try to bridge these gaps. Yeah.
Okay, thank you. And then I would like to read one question which came in. Which solution can we expect from machine learning for genetic variant interpretation of the short run? This is a question from Jirko Kühnisch.
Yeah, I think in the short run, really what I showed in terms of cut is really state of the art. I mean, that is really great and goes far beyond the traditional scoring schemes. And eventually there will be more, but the problem is in genomes, we are really not in a situation of big data because there is so many variants and still comparatively few well interpreted variants that we can learn from. So that is really the challenge there. And but of course this will change also with new experimental methods. Like, I mean, people are now looking into massively parallel assays to see how in organoids or in cell culture things behave, and we can CRISPR things and really come up with big libraries of things and see how they phenotypically manifest different variants, and that is the way forward. And then I think we can expect a lot from this machine learning technology, but it will not all be sequence. I mean, it will also be 3D structure and methylation and so it's probably not over with just the sequence.